Hello everyone, today I will be interviewing Mr. Fagan and Ms. Begum about a recently new discovered form of antibiotics. What can you tell us about this discovery and why is it important?
Thank you, Mr. Simpson. Throughout modern times, antibiotics have become increasingly less effective. So, upwards of 30% of clinical isolates from intensive therapy units and nursing homes are resistant to upwards of 3 or more antibiotics, and millions die to antibiotic resistance each year.
Can we get that image?
Gallium therapy is unique compared to most modern medicine because it specifically targets the bacteria's metabolism and food source. Iron is essential to the survival of the bacteria. Without it, bacteria will die and can no longer replicate. Gallium is a Trojan horse of sorts. It tricks the bacteria into thinking it's iron and starves the cell from within. On top of this, gallium is completely safe for human consumption in certain doses and is FDA approved. It can even prove useful against treating cancer by replacing iron in cells with abnormal growth. Gallium shows great promise as a last resort against normally untreatable antibiotic-resistant bacteria.
That's great, but why is iron important and how does gallium replace it?
Well, like Mr. Fagan said, iron is an essential element for the growth and development of all living organisms, including bacteria and other pathogens. Iron participates in a large number of cellular processes such as ATP generation, detoxification, and cell growth. Iron modulates the expression of the enzyme acetinase via translational mechanisms involving iron regulatory proteins. Acetinase is a vital enzyme for completing the citric acid cycle, and if not expressed properly, the body can no longer finish the citric acid cycle and produce NADH for ATP synthesis through the electron transport chain. It also plays a key role for DNA synthesis, since it is a vital component of ribonucleotide reductase, an enzyme that catalyzes the conversion of ribonucleotides to deoxyribonucleotides. Gallium is a metal that has the ability to exert harmful effects in bacteria by interfering with the metabolism of iron and can hamper all the important processes associated with it. It has a similar ionic radius and a trivalent charge to iron, which can displace the iron in iron-dependent molecules. Because of these similar properties, gallium can also replace iron in important redox reactions, directly hampering the cell's iron metabolism, since iron has different redox chemistry than gallium.
But wait, isn't iron important for us too? Why doesn't gallium therapy affect us?
Well, remember when I said iron was an important part of ribonucleotide reductase and DNA synthesis?
Yes.
Well, since bacteria has such a high rate of replication, this means they will require more ribonucleotide reductase for DNA synthesis, which in turn means they require far more iron compared to our cells, since ribonucleotide reductase requires iron to function. The increase in the need for iron in bacteria means it will attempt to absorb more iron than in the cells in our body. But they are also more likely to absorb the gallium instead. The same thing is true for cancer cells, since they also have a fast growth rate compared to the cells in our body. The gallium therapy can slow the growth rate of the cancerous cells and release the unused iron within them. Once iron is displaced, it can further disrupt the cell by producing reactive oxygen species. Once iron is released from the metalloproteins inside the cell, it is free to react with 2 oxygen atoms and 2 protons to form Fe+2 and hydrogen peroxide. After that, Fe+2 can be further oxidized by hydrogen peroxide to produce Fe+3, a hydroxyl anion, and worst of all, a hydroxyl radical. These radicals are extremely dangerous since they can disable DNA proteins and lipids and initiate cell death.
Okay, well, we know how gallium therapy works in theory. How does it fare in practice?
Gallium therapy has already shown great promise in experimental research. P. aeruginosa is a potential antibiotic-resistant pathogen that can cause deadly bacterial infections in humans if left untreated, can form biofilms that can have long-lasting chronic effects, and is particularly problematic in patients with other illnesses like cystic fibrosis and AIDS. A study conducted with 20 patients, all with cystic fibrosis and similar to and difficult to treat lung infections caused by the P. aeruginosa bacteria, showed that gallium therapy was completely safe on the subjects and significantly improved their lung function while inhibiting the bacterial growth on the biofilms over the course of 14 to 28 days. Another study done with mice infected with the P. aeruginosa bacteria showed that gallium therapy is effective in inhibiting the bacterial growth, and just single doses of gallium nitrate, the drug most commonly used in gallium therapy, was able to cure lethal lung infections. Lab tests also found the P. aeruginosa bacteria were only able to develop resistance to gallium therapy at very low rates, and gallium treatment was even synergetic with other antibiotics. Another study with the mice showed that gallium therapy is also effective against N. tuberculosis. 2 groups of mice were affected with N. tuberculosis, a control group that was given only saline and an experimental group that was given gallium nitrate as treatment. Every mouse in the control group died while all gallium nitrate mice survived, and the CFU, colony-forming units, in the lungs of gallium nitrate-treated mice were much lower than the CFU of lungs of saline-treated mice. It is worth noting that the N. tuberculosis experiment was carried out with a relatively low potency of gallium due to the FDA guidelines. However, a higher potency could potentially yield even better results as new gallium-based drugs are developed and tested. These experimental results are very promising for the future of gallium therapy. As more studies are conducted and new drugs are created, gallium could become a viable alternative to our modern antibiotics.
Is there anything else you'd like to say?
Gallium therapy is unique compared to conventional medicine because it specifically targets its metabolism and food source, and this makes it a viable way to treat otherwise hopeless antibiotic-resistant infections. By tricking the bacteria into using gallium instead of iron, it will disrupt the metabolism of the bacteria and stop it from replicating. With it also being FDA approved for use on humans, it appears as one of the best last resort possibilities of treating previously antibiotic-resistant bacteria.
And there you have it, folks. The future of antibiotics is coming sooner than you think.